So there are new insights into the realm of genetics and epigenetics on aging plasticity. 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 Aging plasticity. The reason that's an important word, plasticity, is because aging is no longer seen as an immutable thing. It can reverse. Okay, so biological aging is characterized by irreversible cell cycle blockade and decreased capacity for tissue regeneration, yada, yada, yada. Throughout the centuries, there are countless examples of humans attempting to escape the inevitable blah, 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 blah. Cellular senescence is one of the important factors that trigger aging, and it is also the most widely studied target of aging intervention. Hayflick first proposed the concept of cellular senescence, and he found that mammalian cell cultures divide to a certain stage, then appear senescent or die, which is known as the Hayflick limit. Mutations in single-cell genes, yada, da, da. The genetics of aging. The lifespans of different biological species lie within a relatively stable range, and there are significant differences between species, which are indicated in the database of animal aging and longevity, as summarized in multiple factors contribute to this difference, including the ratio between body size and heart rate, environmental factors, energy uptake, genetic factors. In terms of genetic factors, whole genome sequencing has revealed that the mutation rate of non-germline somatic cells between a species is an important factor affecting lifespan, yada, da, da. Okay, age-related genes and signaling pathways. Aging is the most important risk factor for a broad array of diseases: neurodegenerative diseases, cardiovascular diseases, metabolic syndrome, chronic inflammation, and cancer. Additionally, genetic mutations that delay aging have been also found to delay the onset of age-related diseases. So genetic mutations that have been found to delay aging also delay age-related diseases. That's not really a surprise. Nutrient sensing. Cells rely on nutrient sensing for both the detection of stresses and ultimately for their survival. Sirtuins are another gene family that can extend the human life. Well, can extend the lifespan of C. elegans, which is a worm. They are mainly responsible for regulating cell metabolism, genome stability, gene expression, signaling, transduction, blah, 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 blah. You can stimulate these in humans as well. We have the same genes. Nuclear skeleton, associated proteins, blah, 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 blah. The production and maintenance of the circadian rhythm is the result of positive and negative feedback loops regulated by a series of circadian rhythm, yada, 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 yada. Disturbances in the circadian rhythm accompany the occurrence of aging and can contribute to the onset and progression of aging. The epigenetics of aging, which is what I want to focus on here. So recent population studies have found that as aging progresses, genetics have a decreased influence on gene expression. Age-associated epigenetics perform a more important role than genetics in determining which genes in the body are expressed, and this affects susceptibility to disease. The relation between epigenetic modification and age has been increasingly apparent, and the impact of epigenetics on health, lifespan, and longevity has been widely studied. Epigenetic changes that occur during aging may not only serve as indicators of aging, but also drive age-associated transcriptional changes to directly affect the processes. Okay, epigenetic processes primarily regulate the aging process. Epigenetic processes primarily regulate the aging process by regulated gene expression via dynamic changes in DNA methylation and histone modification, non-coding RNA, and chromatin remodeling, compared to euchochromatin, blah, 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 histone modifications. So histone modifications, there are numerous possible histone modifications with chromatin Conformation and gene expression being mainly determined by methylation, acetylation, phosphorylation, and chromatin remodeling. Okay, epigenetic clock. DNA methylation levels are clearly correlated with age and can be used to predict chronological age of both blood and certain organs, including kidneys and liver. Thus, this has been termed the epigenetic clock. The epigenetic age of embryonic stem cells is almost zero. It is possible to use algorithms to calculate biological age based on how many sites in an individual's genome bind to methyl groups during the passage of a mouse embryonic fibroblast, blah, 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 blah. This is a little diagram here you're more than welcome to look at. Extracellular vesicles or EVs, mainly involved in the removal of excess or unnecessary substances from cells, the maintenance of homeostasis. So the more your body can maintain homeostasis, the longer you're going to live. Okay? So the more your body can maintain its balance, the better you're going to do and the longer you're going to live. So, RNA, yada, yada, yada. Interventions for aging. So, the rapid progress in the study of the molecular mechanism of aging has made us realize that the speed of aging and length of life expectancy can be intervened by humans. To understand the genetic and epigenetic biological mechanisms behind aging, gain insight into the plasticity of aging, and ultimately achieve effective intervention in aging at present, the most effective aging intervention is calorie restriction, and the development of age-related genetic research has made us realize that aging-related pathways can also be used to as targets for aging interventions such as insulin-like signaling pathways, AMP-activated protein kinase or AMPK signaling pathways, sirtuins, nicotinamide adenine dinucleotide or NAD+, circadian clock, chronic inflammation, cellular senescence, yada, yada, yada. Increasing NAD+ levels can significantly ameliorate aging. So to say that biological aging cannot be halted or even reversed is not an accurate thing. Conclusions: the study of genetic and epigenetic characteristics of aging is essential to the development of targeted treatments against aging. Still, our understanding of the genetics and epigenetics of aging is just the tip of the iceberg. Okay. Now, what I want people to understand, there's a greater likelihood, in my opinion, that scientists and medical doctors are going to be able to intervene and help people with aging and reverse aging than there is that something like counseling or therapy is going to be able to help mental illness. The so-called research behind therapy is very weak compared to the research behind aging pathways. So this is the genetic and epigenetic regulation of human aging and longevity. This is kind of more of the same stuff, but I wanted to be able to explain it a little more in depth here with other people's stuff. So FOXO3. The fork-head winged helix box, group O, transcription factors are crucial components, blah, 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 like growth factor, insulin, IGF-1 signaling, yada, yada. This group was the first to show an association between SNPs and gene FOXO3 with longevity in a study of American men of Japanese ancestry, 95 plus years of age. Since the finding over a dozen independent studies and geographically and racially divergent populations, that's important, geographically and racially divergent populations worldwide have confirmed the FOXO3 finding, with the gene being the next best replicated after APOE, longevity gene association. What I want to prove here is that this is well known. The aging research is far more backed up by reality than is something like what Dr. Grande does, which is therapy. okay? Because I'm not trying to be mean, and I really don't. People know my views on mental health. My view on mental health is highly dependent on subjectivity, highly dependent on a lot of gobbledygook, a lot of psychobabble, and a lot of imagination, okay? Whereas the research done by people like Dr. Morgan Levine, Dr. Kara Fitzgerald, Dr. Walter Longo, Dr. Roy Walford, this is not, their research does not take place in the realm of imagination, okay? Whereas mental health research does. There's very little science in mental health. There's a whole hell of a lot of science behind the reversal of aging. But this is just showing how aging is measured, different genes, yada, yada, yada. Okay, what's behind it? I'm going to move forward here. So this is research that was done by Kara Fitzgerald, who is a naturopathic doctor. Manipulations to slow biological aging and extend human health span are of interest given the societal and healthcare costs of our aging population, yada, yada, yada. So advanced age is the largest risk factor for impaired mental and physical function, and many non-communicable diseases, including cancer, neurodegeneration, type 2 diabetes, cardiovascular disease, blah, 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 blah. Chronic disease is aging. Let's get this straight. I mean, we know that. Dietary recommendations employed as part of the treatment protocol for this study were based largely on biochemistry and generalized measures of health. Because few dietary associations with the DNA age clock have yet been established, a modest blah, 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 blah. So twice daily breathing. So what you want to know, so they use the Horvath clock. This was, I really want people to read the book. What is the book? Kara Fitzgerald's book. Oh my gosh, I forgot the title of the book. But it's a book on how to eat to reverse aging. And it's proven that it works. So the lifestyle guidance, this is what you have to understand. So basically, people talk about blueprint this and blueprint that. So this is essentially kind of like the blueprint, except not vegan. So 30 minutes of exercise per day, at least 5 days a week. 60 to 80% of maximum perceived exertion. Exercise is well known to be broadly beneficial for almost every aspect of health and has been shown to extend mean lifespan in animal models. Exploration of the effect of exercise on methylome has recently begun. For example, regular Tai Chi practice was associated with slowing of aging-related DNA methylation. So it's literally, Tai Chi is literally slowing the aging process, okay? Something that Dr. Grande didn't believe was biologically possible, but guess what? It's biologically possible. These results were not reported in terms of the Horvath clock because it had not yet been developed. One systematic review of human studies found that regular daily physical activity was associated with lower blood levels of homocysteine, and which, when elevated, suggests an insufficiency of methylation capacity. Now, taking B12, folate, and B6 will also lower homocysteine. So keep that in mind. Twice daily breathing exercises that elicit the relaxation response were prescribed for the stress reduction. It was recently demonstrated that 60 days of relaxation practice designed to elicit the relaxation response, 20 minutes twice per day, could significantly reduce DNA M age as measured by the whatever the hell clock, okay? This study aimed to optimize sleep with the recommendation for at least 7 hours nightly, 7 hours in general considered to be healthy, but the limited data on accelerate. Now, my view on sleep is this. People should sleep 8 to 11 hours a night. And people might go, that sounds like a lot of sleep. People that sleep a lot die earlier. I don't mean 11 hours straight up. I mean maybe get a nap or something. Sleep is very important at cleansing the nervous system, particularly the brain, of gunk. okay? And people will go, but people that sleep a lot are at an increased risk of blah, 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 blah. A lot of bodybuilders, people know that I have posted, Frank Zane, it couldn't count as research, but him looking at the lifespan of bodybuilders. Bodybuilders sleep a lot. They sleep a lot because they work out a lot. If you're exercising as much as you're supposed to exercise, or working physically as much as you're supposed to be working physically, you're going to sleep a lot. The other group of people that sleep a lot, that aren't bodybuilders had an increased lifespan, are people that are sick. People that are sick sleep a lot because they're sick, okay? So if you're sick, you're probably not going to have a good lifespan. You got to really pick apart the way the research is done. Okay, D3, blah, 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 blah. Results compared, blah, 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 blah. Let's get to the conclusion. So metabolic measures in blood markers, most significant change was 25% decrease in mean triglycerides over the 8-week period, blah, 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 blah. Discussion. The significance of these findings is multifactorial, but primarily as the first demonstration of potential reversal of epigenetic age in a randomized, controlled clinical trial accounting for any normal variability in epigenetic methylation. This is the second report of diet and lifestyle intervention reducing biologic aging in individuals otherwise known to be healthy. Notably, the shorter time frame of this study and the scale of potential reduction, whilst modest in magnitude, may correlate with meaningful socioeconomic benefits and appears to have the potential to be broadly achievable. Meaning, you can fucking do this. And that's the thing that I think, the one negative I think about Blueprint is people are like, I can't do this, I can't do this. It's not really that hard to do the Blueprint. Some of the things are like the skin stuff and all that, but there are lower cost alternatives to the things that Mr. Johnson implements. Okay? Where the real money comes in is the measurements, but you can get blood tests and things like that done by what do you call it? By the company Life Extension. And if you talk to your doctor and you're very nice, maybe your insurance company will cover an echocardiogram and things like that nature, okay? So you got to take things into consideration. So effects of non-methyl donor factors on DNA methylation, the seminal work of the Waterland and Jertel, in the mouse model, blah, 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 blah. DNA methylation marks as the silence of gene expression and dramatically alter phenotype, blah, blah, blah. Phenotype is, okay. So the genotype is the genes. The phenotype is how the genes are expressed, what you look like, who you are, that type of thing. The power of nutrition to bring about transformative phenotypic changes has held up over the intervening years, most strongly in animal studies. So, if you take people and you train them the same way, like if you train a runner and you put one runner on a high protein diet, okay, and another runner on a low protein diet, the high protein runner is not only going to get less upper respiratory infections, but they're going to get bigger muscles by running, okay? The other guy's going to get real slim, okay? So there was supplementation known for methyl donor nutrients like folate, vitamin B12, choline, SAMe, and betaine, supporting the concept of far-reaching regulatory network on DNA methylation and representing a departure from previous studies that manipulated DNA methylation more directly with extra dietary supplemental... Oh, I'm sorry. Let me back this up here. If I remember correctly, this is why she used meat, was to get the methylation from the meat, from the methionine and the other stuff. So I don't think she used supplements, although in the book she does recommend using supplements if you're a vegan. Rationale for using supplemental methyl donor nutrients. And designing the present blah 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 extra dietary supplementation of methyl donor nutrients was specifically avoided. It was avoided because a growing body of epidemiological evidence indicates potential long-term risks, blah, 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 to which the short-term. She doesn't give good reasons. She's like, might do this, I don't think it's that big of a deal, but then again, I'm not the researcher, and I'm not a researcher, so don't listen to me. Cautions in future directions. One significant limitation of this pilot trial is limited statistical power due to the relatively small sample size. Confirmation of these results is therefore needed in larger study groups. I say, take a whole bunch of college kids and put them on the fucking program. That's what I say. Take a bunch of college kids studying something retarded like mental health, put them on this, and see if they don't switch their fucking major.